I'd like to welcome you to, on behalf of the American Chemical Society and the Agro Division, to this latest in the Lunch and Learn webinar series. Today our topic will be exposure data quality in environmental epidemiology 240 as a case study. My name is Ken Rocky. I'll be host and moderator of this particular session and also helping with questions. And before we go to our two speakers who will be presenting today, I'm going to provide just a little bit of background on the particular product that will be assessed and discussed during this seminar. And so after this brief introduction, we'll go to our first speaker. Let me first make a few introductory comments regarding the herbicide 2,4-D. It's been around for quite a long time. It's one of the first major synthetic herbicides that was introduced, and yet it still continues to be widely used for weed control, both in the US and overseas in various weed control scenarios. It's sold and marketed in quite a large variety of forms and products, and it's got a very robust database of scientific research regarding human and environmental safety as a result of the research supported by the registrants and academic researchers. And it's currently proved in around 100 countries, including in the US, where the Environmental Protection Agency is the evaluating body. Human exposures can occur to trace residues from both occupational and non-occupational scenarios, so people working with the product, pesticide applicators, workers re-entering treated fields or areas, and also residential or non-occupational exposure through, for example, trace residues in food and water. As mentioned, 2,4-D continues to be a very important product both in agricultural and specialty use weed control scenarios. It currently ranks by volume number 1 as far as use in home and garden areas, number 2 for industrial vegetation control, and number 4 for agricultural uses. And you can see from the chart on the right of the slide some of the major uses in terms of pounds over a period of several years, with some of the important crop uses being things like cereals, pastures, corn, soybeans. But you can also see turf and residential uses as being, again, significant use area for this particular product. With that very brief introduction then to 2,4-D, the herbicide, let's move on to our speakers today. And we're happy to have 2 symposium lectures from an Agro Division symposium organized as part of the 254th National Meeting in Washington last year for an encore performance. We'll have the 2 speakers, and then we're going to hold questions that you may submit through the question bar until the end of both speakers. And again, Ken Rocky here, I'll be moderating those. Let me introduce the first speaker then. That's going to be Dr. Carol Burns, and then later I'll provide a few more details on our second speaker, Dr. Judy LaKind. But first, a few words about Dr. Carol Burns, who is President of Burns Epidemiology and Consulting, also an Adjunct Professor of Epidemiology at Central Michigan University College of Medicine. She's worked for a number of years in the area of occupational and environmental epidemiology at the Dow Chemical Company, and her primary areas of focus are assessing exposure in epidemiology studies and improving the use of epidemiology for use in risk assessment. So, Dr. Burns.
Thanks, Ken. We often hear that, oh, chemicals are tested with animal studies, but why don't they use epidemiology studies? Because they're the most relevant. And in fact, epidemiology is used for decision-making in public health protection. And I'd like to give an example of 2,4-D because it's been around so long and tell the story of the fits and starts that it's had with being used for public health decision-making. But before I go too far, I'd like to give a few definitions that are relevant for epidemiology. And this is mostly about how we conduct our studies. The first is the case control study. And this really gets at how we identify our participants. So you imagine the cases are people with disease and our controls are those without. So this might be, we hear this in the news when there's a salmonella outbreak. And you want to identify many possible food items that they might have eaten, and you compare that to people who didn't get sick. Another design is the cohort study. And this is where we want to focus on a single exposure. So this might be a manufacturing group, in this case 2,4-D, or some other group, if you will. And then you can follow them and look at very many diseases. So it depends on the focus of your study, of which type of study that you'll do. The third type of study is called the ecological study. And this is probably the weakest design, but it's really good for generating hypotheses. And sometimes you might hear this used in the news if you're comparing different countries, if you will, maybe Japan, United States, and France, and compare their cancer rates and make inferences based on their diet of why one country might have a higher or lower cancer rate. As you can imagine, this is not a very strong design. Lastly, I use dose very loosely. A toxicologist or a physician might use dose very specifically, but for me and in this presentation today, it's just an estimate. So let's get to the beginning of our story. Early in the epidemiology world, we really focused on occupational groups. For the most part, this is because they tend to have the highest exposures and the most easily identified. In the case of 2,4-D, it's really been the standard to ask individuals using a questionnaire about their exposure. What did you do? How many years did you do it? And in an average year, how many days did you apply this pesticide? And I'll show you how this looks in the case of a case control study. So back in the 1970s and 1980s, non-Hodgkin's lymphoma and soft tissue sarcoma were really cancers of interest. What was causing these cancers? Another big interest was the dioxin contaminant of some of the phenoxy herbicides. So in 1986, the National Cancer Institute published this study they had done in the largely agricultural state of Kansas. And I'll just show you how to interpret this slide because I'll have several like this. So if you can imagine, if your cases, your people with cancer, and your controls, the people without cancer, had the same experience with exposure, their risk estimate or odds ratio would be the same. It would be 1. And so this is our line of no risk. And so you look to see if the odds ratio or our risk estimate for a case control study falls below or above the line. And here for this study, we see these odds ratios are all above the line, so suggesting increased risk. We also interpret the role of chance and random error with error bars. In this case, they're called confidence intervals. And essentially, we look to see if this confidence interval crosses our line of no risk. And we say that when it does, we interpret that odds ratio, and here we see at the highest dose, odds ratio 6 to be statistically significant. And I'll introduce you down here at the x-axis. This is our dose. In this case, they asked a question, how many days per year did you use herbicides? So we have an increasing dose and looking to see a trend almost as they used more herbicides in a given year. Now, as you can imagine, the reaction to this was dramatic, and the US EPA put 2,4-D under a special review. They issued a data call-in, which essentially required more toxicology studies relating to cancer and more analytical chemistry to the contaminant. Commercially, a national lawn care company deselected 2,4-D from use. And as always, more research is needed. So the National Cancer Institute replied by doing another study in the state of Nebraska and improving their questionnaire. So this is what the Nebraska study looked like. Again, we have this odds ratio here, and we see an apparent increasing risk with increasing frequency of use or dose, but this time it's not statistically significant, even though they've collapsed the doses into 3 groups from 4. And this is an important time to stop and talk about case control studies in general. They have a problem with them called recall bias. And it's human nature, if you can imagine, that if you had cancer, you're thinking a lot about what might have precipitated that getting cancer. What had you done in the past that might be related to getting cancer? Whereas the persons without cancer, the controls, who are just contacted randomly on the phone, they may not have that same thought process. And as a result, sometimes you have this bias introduced or this error in which the cases and controls report the same experiences differently. Another problem with case control studies is another type of bias related to the type of respondents. And here you see the results that were different when the reporting were self-reported. What I mean by this is, again, when you're contacting individuals with cancer, sometimes by the time the investigator has reached out to that individual, they're too ill or they've died and cannot participate in your survey. However, the investigator doesn't want to lose that information, and as a result, they interview either a family member or another household member who would know about the experiences of that person. But what we see here is the odds ratios are very different for self-reporting versus the proxies. So what to do? Well, the 1990s saw a lot of activity with 2,4-D in epidemiology. There were more studies in Iowa and Minnesota, Washington, and New Zealand. And the US EPA, in evaluating these studies, essentially said, this isn't sufficient. We can't use this information. It's not good enough. The US EPA also reverted back to, we need some more robust studies on the rat and the mouse. In reply, the National Cancer Institute then said, okay, we're going to do a cohort study. One of the largest cohort studies initiated. Almost 90,000 applicators and spouses in Iowa and North Carolina, looking at, again, enrolling them before they have disease so that the investigators could follow them forward to see what diseases presented. So they didn't have to focus just on non-Hodgkin's lymphoma or even just on cancer. And here I'm going to stop sort of the middle of our story, if you will. So most of these studies were relying on questionnaires. And you can, even if you have a really good question, such as, do you own running shoes? It doesn't necessarily get at the question the answer you really want is, how fit am I? So there were some efforts in the late 90s and early 2000s to start to validate some of these agricultural questionnaires. And here's an example of a study that was done in Canada. And they enrolled 126 pesticide applicators who were either applying 2,4-D or the other herbicide, MCPA. And they collected urine before, during, and after the application, looking to see, well, if they applied 2,4-D, what were the levels of 2,4-D in the urine? And as you can see, it was about 50-50. About half of the participants didn't have any detectable traces of 2,4-D in their urine at all. And the levels were better predicted by behaviors such as handling practices, personal hygiene, and their protective clothing. So what we learned about exposure was not necessarily what we expected. There was a lot of variability both between and within individuals. Some applicators didn't have any pesticide traces in their urine. And if you think about if you've ever sat next to somebody on an airplane when they're eating, you know that there's a big difference in behavior and how neat and tidy some people are or are not. We also learned that quality matters. It really is important about how you collect the data, and Dr. LaKind will talk further about this. And lastly, there's no gold standard. Both questionnaires and biomonitoring are important, but collecting both pieces of information is really the better way to go. So let's fast forward. Here we are today. There have been more studies, more reviews of more studies, and several meta-analyses of these reviews and these studies. And what have we learned? Well, first of all, the results are mixed. And just like the squeaky wheel gets the grease, often some studies get more attention than others. This time on this graph, what I have at the x-axis are the publications in publication order. And you can see there's some here that are statistically significant, and we often hear about those in the news. But for example, they're not that strong. Here, this one was an ecological study. They didn't have exposure information on individuals. Another study, not statistically significant, showing hardly any increase at all, is a manufacturing cohort, one of the few that wasn't reliant upon questionnaires. Remember we talked about the Ag Health study? Here are their results, no increased risk at all. And lastly, here's the meta-analysis that was recently published, which is basically a mathematical sum of all these studies put together. Again, showing no increased overall for 2,4-D and non-Hodgkin's lymphoma. The regulatory interpretation of 2,4-D using these epidemiology studies shows the same conclusion. In 2008, the Canadians, PMRA, recognized that yes, there are some associations of non-Hodgkin's lymphoma and pesticide use, but they aren't consistent. As we speak, the US EPA is evaluating 2,4-D, and they came out more definitively saying no clear associative or causal relationship was observed. What this means for epidemiologists is that we really need better guidance if we want our studies to be used and relevant in these regulatory decision makings. The US EPA put out a framework for using epidemiology and risk assessment, and they recognize that they're going to start using study quality, not just doing the mathematical assessment of all those studies. And in fact, some low quality studies may not be included. Our counterparts in Europe, the European Food Safety Authority, also proposed recommendations on improving both the quality and reliability of epidemiology studies. They came out pretty strongly saying that good quality studies were rare, and that it was difficult to harmonize exposure information across studies. For example, if you're looking at a manufacturing study where the individuals are exposed possibly daily compared to farmers or applicators who may have more episodic exposure. And lastly, stating that good studies may be let down by the poor reporting of the information. With this in mind, epidemiology results do get noticed by regulators. But keep in mind, too, that pesticide regulation is extensive and data-rich, so epidemiology may just be a small piece of that decision-making. Poor exposure characterization may be the reason why we see these vast inconsistencies across epidemiology studies. And as an epidemiologist, we need to have frank conversations about this, and are we ready to rise to the challenge so that our data are more relevant? With that, I'm going to turn over to our next speaker, Dr. LaKind. And if you have any questions for me, please put them into the chat function, and we'll answer questions at the end of the session so that we know we have plenty of time.
All right, very good. Well, thanks so much for your presentation, Dr. Burns. I think that's been a good kickoff to our session, and we're going to move on then to our next presenter, Dr. Judy LaKind. Yeah, Dr. Judy LaKind is President of LaKind Associates and also Adjunct Associate Professor in the Department of Epidemiology and Public Health at the University of Maryland School of Medicine. Dr. LaKind is also President of the International Society of Exposure Science. Dr. LaKind is a health and environmental scientist, and her expertise is in the area of exposure science, assessment of human health risks, biomonitoring, scientific and technical analysis for regulatory support, and state of the science review. So Dr. LaKind, welcome.
Thank you so much, Ken. So I wanted to start first by thanking the Agro Division of the American Chemical Society for inviting me to participate in this webinar. And I'm going to be building on the information that you heard already from Ken Racky and Carol Burns by focusing on the exposure component of studies on 2,4-D exposures and human health. And in particular, I'll be describing a process for assessing quality of exposure data, and then discussing the results of research that my colleagues and I conducted using this process. So this slide shows my colleagues who were part of this research. You can see Carol Burns, who you just heard from, Dan Naiman, Cian Omani, Julia Vallone, Annie Byrne, and Josh Naiman. And I'd also like to note that the research was supported by the Industry Task Force on 2,4-D research data, and that they weren't involved in the research or the publication process. Okay, since you already heard some background on 2,4-D from Ken and Carol, what I'd like to simply say is that 2,4-D has been in use for decades, and along the way, scientists have collected a lot of information on concentrations of 2,4-D in various environmental media and also in humans, mostly for the latter by measuring 2,4-D in samples of urine. Each approach has strengths and limitations, but the quality of the data from both approaches can be assessed. And just to be clear, when I refer to data quality, I'm also talking about the level of confidence that we have in our data. So why do we need good quality exposure data? Well, we use concentration data from environmental media and humans to characterize and classify exposures, both in epi research and in human health risk assessments. And it's important to remember that the quality of the exposure assessment is actually a major determinant of the overall quality of any environmental epi study. So if we're trying to understand whether there's an association between chemical exposure, shown here on the x-axis, and a response or health outcome, shown here on the y-axis, then we need to be sure that we have good quality information on exposure, because if we don't, our ability to link an exposure to a response will be hampered. So the goals of our research were to examine the published 2,4-D measurement data from both environmental media and humans, and we wanted to be able to answer just a few questions. We wanted to be able to ask or answer, what can the data tell us about sources and timing of exposure to 2,4-D? In other words, do the measurements represent peak exposures or chronic exposures or maybe something else? How generalizable are the data? If we have a study of 2,4-D measurements in urine from a group of children living in, say, Ohio, can we use that information to say something about exposures to children elsewhere, or can we use it to say something about other age groups? And then finally, what can we say about the quality of the data? So we started with a literature search with the aim of capturing all published studies with 2,4-D concentration data. And we identified 241 studies that had measurements of 2,4-D in food, soil and dust, air, water, urine, blood, and semen. And you can see in this slide the number of studies we reviewed. That's in that middle column. We excluded studies from our review for a number of reasons, and I won't go into that here, but just as one example, if a paper was focused on human health effects from 2,4-D, but didn't have any concentration data, for example, like the studies that Carol mentioned where questionnaires were used, then we didn't include it. So after our screening of the 241 papers, we were left with 156 papers to evaluate, and you can see the breakdown by medium in this slide. So how do we assess quality? This is one of those words that means different things to different people. In order to provide consistency to the underlying definition of quality, in 2014, a group of scientists developed a published instrument that can be used as a guide for conducting a comprehensive and transparent examination of quality of exposure information in environmental epi research. And we called it the Biomonitoring Environmental Epi and Short-Lived Chemicals Instrument, or the BC. But it actually has a wider applicability. It can also be used for persistent chemicals, and most of the elements in the instrument can also be used to evaluate not just the quality of human data from biomonitoring, but data from environmental media as well. So the instrument has some important features. For example, it doesn't score studies, but rather it uses a heat map approach, and that makes it easy to get a feel for the overall quality of a given study. And the figure here shows a hypothetical example where the quality elements, which I've not yet told you about, are in the left-hand column, and then for each element, we decide based on specific criteria whether a study is top tier, and that's shown here in green, or a lower tier, shown in yellow or red. So the instrument provides us a way to evaluate various aspects of study quality in a transparent and reproducible way. Now, I don't have time in this talk to discuss all of the quality elements in the BC. What I show here are the 4 elements of study quality that we used in our research on 2,4-D. So we were focused on whether the researchers demonstrated avoidance of sample contamination, and analyte stability, how they dealt with matrix adjustment, and also variability. Now, because of time constraints, I'm just going to focus on 2 of these. First, I'll describe issues around whether or not the sample Either environmental or biomonitoring was kept free of contamination. And then next I'll talk about stability of the chemical or the analyte in the sample. So contamination of the sample by the analyte of interest can happen anytime, from the collection of the sample through storage of the sample all the way through to analysis. And while it may seem like avoiding sample contamination should be relatively straightforward, this actually can turn out to be a difficult task in many cases. Just to highlight this, I'll give you an example that comes from research at the Centers for Disease Control and Prevention, or CDC, which has state-of-the-art labs and highly trained personnel. So this example has to do with measuring an antimicrobial chemical that you're probably familiar with called triclosan in urine samples. The CDC lab went to great lengths to make sure that triclosan wasn't present in the laboratory so that urine samples wouldn't be contaminated during the analytical process. Yet during their analyses, they started to detect sample contamination anyway. As it turned out, this was due to an analyst who was eating lunch in another building that happened to have a restroom with hand soap that contained triclosan. And then they would go back to work, and that's all it took to introduce triclosan into the urine samples. In a similar event, triclosan contamination of samples was found to be caused by an analyst who was using triclosan-containing toothpaste in their home. And CDC also found that chemicals called benzophenone 3 and parabens were contaminating lab samples, and this time the chemicals were introduced into the lab by an analyst using sunscreen and underarm deodorant containing these chemicals. So even if your laboratory is extremely clean, a simple change in behavior by a chemist at their home can produce sample contamination. Now, things get even more difficult when we start talking about field studies. So imagine a situation in which you want to collect soil samples from various parts of a field, and it's got differing levels of chemicals from location to location. You need to be sure that your sampling equipment has been thoroughly cleaned in between collections, and that's not really an easy task. And similarly, imagine that you want to get a urine sample from a person who's been spraying a pesticide during the day. So you might leave a clean sampling cup for them to use, but you need to know that they'll be able to clean their hands and other body parts sufficiently so as not to contaminate the urine sample. So the take-home message from all of this is that epi studies that rely on measurements in human matrices or environmental samples, they need to demonstrate the steps they've taken to avoid sample contamination, all the way from collection of the sample, through handling of the sample during shipping and storage, and all the way through to analysis. So the highest quality, or Tier 1 study, will include that documentation. In a Tier 2 study, maybe there's incomplete documentation, but in a Tier 3 study, there's none at all. Okay, so now we can evaluate the 2,4-D exposure literature, get a feel for how we're doing. So let me get you oriented with this slide. The left-hand column shows the publications we reviewed sorted by medium and also includes the number of papers for each medium. In the right-hand column, you can see a pie chart for each medium. So let's start by focusing on the green segments for each pie chart. And just as a reminder, that refers to the number of studies that are Tier 1, or the highest quality. So for example, at the top, you can see that for foods and beverages, we found 7 publications. Of those, 5 included documentation of avoidance of sample contamination for both field and lab components. But in contrast, if you look down at the urine samples, we found 52 papers, and of those, only 3 were Tier 1. Now, by glancing down the entire column and focusing at the red or Tier 3 segments, you can see that many of these papers had no documentation at all regarding whether they kept the samples free of contamination. Now, it's worth pointing out that this doesn't necessarily mean that the researchers didn't do this, but if they did, the steps weren't documented, and from reading the paper, we have no way of knowing if any necessary steps were actually taken. So the other element that I'm going to be discussing today is analyte stability. So samples are generally collected, shipped to a lab, and then they're frozen until such time as they can be defrosted and analyzed. Samples can be stored for years prior to analysis. Sometimes samples are frozen, and then they're partially thawed, and an aliquot of the sample is withdrawn for analysis, and then the remainder of the sample is refrozen, and this can happen many, many times. So a lot of time can pass with varying storage conditions that may affect the chemical that you're interested in. the concentration of the analyte in the sample then can change due to things like degradation or volatilization or even absorption to the walls of the sample container. So as an example, you can see here the results from a study by McManus et al. And along the y-axis are concentrations of several analytes measured in groundwater, and this includes 2,4-D, and the x-axis is time in days. And the red curve, and I added the red to this figure, this is for 2,4-D, and you can see the loss of 2,4-D in these samples. This is about over a month, but this far gets you over about a month, for samples that were stored at 4 degrees centigrade or 40 degrees Fahrenheit. So what should be clear is that researchers need to demonstrate that the concentration of the chemical that's being reported reflects what was in the original sample. So samples shouldn't be stored beyond the time frame of demonstrated stability of the analyte. So really simply put, for a Tier 1 study, publications should include information that shows that the analytes they're interested in are stable from the time the sample was collected through the time of sample analysis. So this slide is similar to the one I showed you on sample contamination, but now we're looking at documentation of analyte stability. In this case, you can see that for the 7 papers that we found for 2,4-D in foods and beverages, none demonstrated analyte stability, nor did they provide information on sample history. And actually, if you look down the column of pie charts, you can see that really very few papers are Tier 1, or it would be green. In fact, you can see that most of these papers didn't include sufficient information on sample history or analyte stability that would ensure that their concentration data are accurate. Now, again, it's worth pointing out that this doesn't necessarily mean that the researchers didn't address analyte stability, but from reading the papers, we have no way of knowing. So, what are the take-home messages? For avoidance of sample contamination, our review found that few publications, only around 20%, documented steps taken to avoid sample contamination in the field or the lab, for example, by using things like field blanks and lab blanks. So this leaves us with concerns for internal validity, because contamination can result in falsely elevated concentrations. But there's a pretty feasible fix here. We know how to take steps to ensure that our samples are free of contamination. And there are guidance documents that are readily available to assist researchers. So we need to use and document these procedures. And in today's publishing environment, I think it's much easier to include longer method sections because we have the ability to provide supplemental material with our papers. For analyte stability, we found that even fewer papers, in this case only around 2%, demonstrated stability of 2,4-D over the time from sample collection to analysis. Most publications didn't report on sample storage times and conditions. So again, we have a potential issue with internal validity of these studies because things like analyte degradation or loss due to any mechanism can result in falsely lower concentrations. So the easy part of the fix here would be for researchers to document storage times and conditions and provide information on the stability of the analyte. However, it's not really clear how many researchers actually study analyte stability for their research conditions, and this is a situation that may need to be improved. So to conclude, we found 156 publications that had data on measurements of 2,4-D in various media, and it's important to note that we shouldn't be thinking of this as one big database. Now, I didn't go into this today. We have to consider the fact that there have been temporal changes in use and in formulations and in analytical methods for 2,4-D. So comparing across studies is an exercise that you would have to undertake with great care. The second important point is that not all exposure data are created equal, and any assessment of human exposures to 2,4-D has to consider data quality. Third, I'd point out that there is increased interest in using human data for regulatory decision-making, and so the demand for high-quality exposure data is going to grow. So this is an area that we need to be paying attention to. And then finally, the BC instrument provides a mechanism for conducting transparent and reproducible assessments of quality. But we also have to be asking, what constitutes data of sufficient quality? How good is good enough? And this relates to, are our data of sufficient quality for a particular purpose? It's often referred to today as fit for purpose. And this is a conversation that's begun between scientists and regulators, and it's a conversation that needs to continue. So I'll close with citations for the two papers that were discussed during this presentation for those of you who want to take a deeper dive, and I thank you for listening.
All right, thanks so much to Dr. Burns and also Dr. LaKind for their very interesting presentations regarding exposure data quality and environmental epidemiology with 240 as a case study. We're able now to take a few questions. So again, those can be submitted through the question tab that you have. And again, we got a few minutes for questions. I see one has come in here for Dr. LaKind. Maybe we'll start with that one. Dr. LaKind, this BC instrument you described was very interesting, and you showed how it was applied to 2,4-D, a very commonly studied pesticide product. Have you used this instrument for other pesticides or other type of materials and found similar things or differences?
So thank you for the question, and we've applied the instrument to a number of chemicals as part of a series of systematic reviews, including one for triclosan and one for bisphenol A, actually two for bisphenol A, and other chemicals as well. And some other organizations are beginning to use the BC for their assessments as well. So it is increasing in use. And as an aside, I'll just mention that we have finished adding another component for model data. So we hope to continue to expand the instruments so that it covers exposure information more broadly, not just for measured data.
Hey, Ken, this is Carol. If I could add to that as well. Dr. Tim Pasteur and I used a modified version of the BC to review the pyrethroid literature, and that's a new publication that you can access as well.
All right, very good. Yeah, again, continue to submit your questions through the question tab, if you will. We've got one question online here. Have reviews of study quality gone through the process of requesting additional documentation that may not have appeared in the original publication? I guess, Judy, that would be for you.
Yes, that's a really interesting question as well. So we did not do that step as part of our assessments. The thing that I would say about that, because we pondered this, so there are some studies where you could go back and find the researchers and ask them that question. A lot of the studies we looked at were quite old, and I don't know how many people saved their laboratory data and methodology from the 1970s and 80s. And I was thinking about this because I was thinking about a study that we did in that time frame, and I was thinking, I have no idea where those notes are at this point. And so I think we need to be focusing in this as we move forward. Any information we can get historically would be great, but I think we'll be doing ourselves a favor if from this point forward we start including that kind of information so that 10 years from now, people will understand the quality of our data and they won't have to track us down.
Yeah, thanks so much. There was some discussion at the start that dealt with how regulatory agencies view some of the information that's been published and specifically regarding sort of the documentation and the quality of the analytical and exposure pieces. Is there any sense? I mean, are the regulatory authorities starting to take this more fully into account? I mean, do you have any examples where you've seen this incorporated into? Because again, there seems to be an emphasis on use of such published information in the regulatory process. But how about this question of reliability? Do you see this being addressed? And I guess maybe we'll start with pesticides.
Carol, do you want to start?
Sure. Well, it's the kind of thing that everybody wants, but do it with somebody else's data. Sharing more results is really certainly important, and I know that NIH is coming out with recommendations for studies that they fund. There are some journals, such as the +1 journal, that is asking investigators, when you publish in our journal, we want you to share all your tables and your raw data. But that's not to say that not everybody is willing to share their data. And if you can imagine it, it does have some ramifications with I'm not done yet. I want to publish some more. I don't want to get scooped. So I want to hold my data and analyze it. And then, of course, there's always that concern that, well, I'm a little nervous that you might find a mistake and then we'll get caught in this loop. So there's discussions ongoing, and I would say the example, Ken, is certainly with chlorpyrifos right now and neural development effects. There's a number of cohort studies that are under discussion, and the question is, can those investigators and will those investigators make their data available? So it's certainly a topic for conversation, but as Judy mentioned, I think we'd all do ourselves a favor by, in the publication process, submitting as much information as we can in the supplemental materials.
Great. Thanks so much for sharing on that one. Here's a question regarding a body we've been hearing more about the last several years, and that's the WHO's IARC process, International Agency for Research on Cancer. And the question here is, to our knowledge, does IARC apply anything like this BC model to the studies they're relying upon for their determinations of cancer classification?
So I don't believe they do. Where we've seen it discussed and at least partly used so far is with the EPA's Office of Pesticides and the European Food Safety Authority. And we're working with a group that's developing an instrument to evaluate risk of bias in observational epidemiological research. And it's likely that it will be incorporated into that process as part of the exposure assessment evaluation. And we have another effort going on right now to develop a broader, I'll call it a value for risk assessment instrument for trying to understand what we can extract from environmental epidemiology for decision making. And that would cut across an evaluation not just of exposure, but also of population and comparator group and outcome. And the BC and its expansion would be pulled into that as well. So there are a lot of, and I should have mentioned also, NIEHS has also discussed the BC as part of its systematic review guidance. So it's the focus on evaluating quality of exposure is being pulled into a lot of different tools and places.
Great. Yep. Thanks for summarizing that. That is an encouraging development. Dr. LaKind, one thing you mentioned was this topic of variability, and a question is, how does the variability in concentrations of 2,4-D in your case study affect our understanding of these 2,4-D exposures?
Yeah, that's another great question. Why don't I start, and then Carol, I know you've thought about this extensively, but variability is a huge issue, not just for 2,4-D, but for many, if not most, of the chemicals that are the focus of attention today. So in the day when we were all expending our resources on chemicals like dioxin, PCBs, DDT, we didn't have to worry about this as much because these chemicals persist in the environment and in humans for such a long period of time. A sample taken today is probably reflective of the concentration in that sample a year ago or 2 years from now. The chemicals that we're looking at today, the 2,4-Ds and triclosans and bisphenol A's of the world, have extremely short half-lives. And so we are still oftentimes in our studies collecting one sample and hoping that that one sample will represent longer-term exposure. But I think there is a growing recognition that that's not the case. And that is really problematic in terms of interpretation of a lot of the research that's going on now and that is already published. Carol, do you want to add to that?
Sure, thanks, Judy. To add to what Judy was talking about, again, looking at comparing environmental monitoring data over time, it's really, you often hear studies or accounts in the news that X% are detected in urine or whatever media. But we have to be careful with that as our metric, because the limit of detection in the last 20 years has gone down to a tiny amount I can't even fathom. And, you know, when you start with a part per million and you go down to a part per billion and lower than that, then when you're talking about detecting, that's very different than what we were talking about 20 years ago. So again, looking at analytical chemistry and advances, we need to advance our interpretation at the same pace of our analytical skills.
Great. Well, thanks for sharing those insights. Dr. Burns, back to some of the information you were presenting. Some of the specific data you were sharing seem to be related to this NHL, non-Hodgkin's lymphoma. And for your 2,4-D example, any other comments to make on other cancers that might have been examined or non-cancer diseases? Were some of the same things applicable there?
Sure. Well, as I started out saying that a lot of the research has focused on non-Hodgkin's lymphoma because of the design of the study, that you start with that disease. But there have been some other studies that have looked at all ranges of cancer, you know, from head to toe. And for the most part, those studies have not shown an association with 2,4-D and cancer.
Okay. Thanks very much. Here's a question, more of a general question about the study of 2,4-D, and that is, are toxicology studies with live animals still going on with 2,4-D? Is that still allowed? Are we still doing animal studies with 2,4-D toxicology? Maybe
well,
Dr. Burns.
Sure, I'll start that, and maybe Ken, you want to wrap it up. But to my knowledge, that they still do studies, but for the most part, they're more mode of action and have very specific questions related to enhancing our knowledge about how 2,4-D is metabolized in the body and so forth.
Yeah, good question. And again, Ken Rock here, I can weigh in a little bit, and that is we've got a tremendous database of historic studies, animal toxicology studies with 2,4-D, and I think in most cases, you know, it's certainly sufficient for current assessment questions, endpoint selection. As guidelines evolve in some regions, however, authorities do call for new data, call for new studies, particularly as review programs go on, where older products like 2,4-D are again examined. And so there are still cases where regulatory authorities feel that, again, some additional, more specialized study may be needed. In some cases, there might be a 20 or 30-year-old study that may not have full information that it is felt that should replace with a modern one. And I think there's a lot more concern and attention today paid to questions related to animal welfare, and if we do testing, do we really need to do the testing? No kidding. Are there alternatives? Are there assays, receptor studies that can be done? But some studies are still done, again, generally where required and absolutely necessary. Yeah, here's a question here regarding epidemiology studies, and the question is, are epidemiology studies intended for use in regulatory decisions required to comply with EPA's human studies rule? And Dr. Burns, this might be a question for you.
Right. The short answer is yes, but as with most things, there's more involved. Most human studies do go through a human studies review board at their own institution first. And then I know from a study we did when I was at Dow, we submitted our protocol to the EPA Human Studies Review Board, and they gave us a pass and said, okay, you've met our criteria, so you don't have to submit any more information to our review board. So that's about the extent of my knowledge on that.
All right, thanks for mentioning that. Carol, maybe a more general question again for you, Dr. Burns, would be what would you recommend as far as new work being done and how best to pursue evaluation of exposure to 2,4-D in an epidemiology study? What things might you focus on?
Well, I think it's really important to, like I tried to iterate before, not to rely on any one single exposure metric. So for example, questionnaires are really good to help us understand what was going on in the past. But I think questionnaires should be matched with, say, a biomonitoring effort to make sure that you're asking the right questions and that the questions are relevant to the information that you're wanting to obtain. So I think it's important that before we start looking at, is there a health outcome, how well can we evaluate and discern one exposure from another?
If I could just add to that briefly, the other piece that I would just bring up is that there's really nothing that replaces being in the field. And I was thinking about a story that a colleague told us about. It was a questionnaire-based study on farm workers, and one of the questions was, do you own personal protective equipment for pesticide spraying? And the answer was yes. And so the assumption would have been that they were using it. And yet when they were visiting the farmer and they looked around, the farmer was going out to spray, and there was the protective equipment hanging in the barn and just untouched. And so the question wasn't really getting at the actual behavior of those farmers. And so again, a site visit and some actual field work, I think, is critical for these kinds of studies.
And to circle back to that, you know, one thing that we always hear about in epidemiology studies is confounding. And if you think about it, again, what is it about that guy that bought the equipment, but it's still hanging in his barn? And the question we want to know is, so he may have higher exposure to whatever it is he's applying, but based on that very basic behavior that he has equipment that he's not using, you want to know also what are his health and his risk-taking behaviors in addition to that. Does he go to the doctor as frequently as he's supposed to? Does he take, you know, his heart medications and so forth? And one behavior is also often a predictor of another behavior. And this is where epidemiology studies often fall apart, is that we assume that a high exposure of whatever it is we're measuring is related to disease. But what we really need to think about and be creative in measuring is that behavior that leads to exposure also a behavior or a trait or a practice that leads to poor health? And that is both the fun part and the difficult part of epidemiology.
Yep. No, thanks so much. Yeah, maybe one last question here. This is about, again, assumptions about exposure scenarios, and the question is, for occupational hazard exposure, if indeed proper techniques are being used by the applicators to limit exposure, what would we still predict there to be higher exposure among that type of a population versus somebody just randomly selected who is not involved in, say, application or handling?
That's an interesting question, and it's certainly one that you know, we've been facing for decades. That you know, you start with the manufacturing population, and if they're wearing their proper gear, should we really be concerned about individuals outside the fence line? Then we start, if our applicators are protected and using good practices, do we need to be concerned about individuals within the realm of drift and downstream and related to that point. So I think it all gets back to our ability to test our assumptions. If that is indeed our assumption that we've protected our applicators and now do we need to be worried about the schools nearby, well, let's ask that question so that we can design the study to test our assumptions.
Yep, very, very, very good. All right, well, it looks like our questions are tailing off and our hour is just about done. So again, I'd on behalf of the ACS Agro Division, I'd like to thank Dr. Carol Burns, Dr. Judy LaKind for presenting this encore presentation regarding exposure data quality and environmental epidemiology 240 case study. Again, we'd like to thank all of you for participating in this latest Agro Lunch and Learn webinar series. Special thanks also go to our webinar sponsor, EAG Laboratories. And please check the AgroDiv website for coming attractions through the Lunch and Learn webinar series. And we thank you so much for all of your participation in today's session and wish you a good day.